good morning, good afternoon, and good evening. My name is Dr. Gulfaraz Khan. I'm Professor of Viral Pathology in the Department of Medical Microbiology and Immunology at the College of Medicine and Health Sciences, United Arab Emirates University in Abu Dhabi. I would like to welcome all our attendees to today's webinar. We are honored to have His Excellency Mirza Sayegh representing His Highness Sheikh Hamdan bin Rashid Al Maktoum, Minister of Finance and the Patron of the Sheikh Hamdan Award, and His Excellency Ahmad Al Hashmi, board member of the Hamdan Medical Award. Before I introduce today's topic and the speakers, I would like to invite His Excellency Mirza Al Sayegh to introduce this webinar series. Your Excellency.
Okay, can you hear me, Doctor?
We can, thank you, yes.
I will start again.
I'm very well, thank you. Welcome.
Thank you very much. Thank you, Doctor, for the introduction. I wish I can be with you. Just calling on ladies and the gentlemen who are present with us today. I'm very pleased to welcome you all to this webinar series organized by the Center for Arab Genomic Studies, a division of the Sheikh Hamdan bin Rashid Al Maktoum Award for Medical Sciences. This is the first webinar of the series that will cover diverse topics around the human genetics and genomics. With the organization of this webinar, the Center and Shams Magazine continue the tradition of working towards raising the awareness and sharing the knowledge about the field of human genetics. These programs are made possible only due to the continuous support and patronship of His Highness Sheikh Hamdan bin Rashid Al Maktoum, the Deputy Ruler of Dubai and Minister of Finance, and our patron here at the Medical Award. I would like to extend on all behalf of all the present my appreciation to His Highness Sheikh Hamdan for his generous support and encouragement to medical research, not just regionally, but also internationally. I would like to take this opportunity once again to thank the speakers, the organizers, and the moderators who have taken time off their busy schedules and agreed to be part of this webinar series. On your behalf, I would like to raise the issue of thanking His Highness for his support, and also on behalf of Sheikh Hamdan, I wish you all a fruitful and educational webinar for the benefit of the humanity as a whole. I thank you very, very much for the introduction and hope that your deliberations and discussions and above all the speeches, the reports will be available to humanity. Thank you once again and wish you good luck.
Your Excellency, thank you very much for your kind words. Let me introduce the webinar of today's session. This is visible? Yeah. Okay. Today's webinar is on genomics of infectious diseases, and we have two speakers, Dr. Youssef, Idaghdour, and Dr. Ahmad, Abou Tayoun. The first speaker will be, let me move the slide. The first speaker will be Dr. Youssef. Dr. Youssef is an assistant professor at New York University in Abu Dhabi. He did his undergraduate studies in Morocco and his master's in molecular genetics at the University of Leicester in the UK. He then pursued his PhD studies in genetics at the North Carolina State University. This was followed by postdoctoral fellowship in medical and population genomics at St. Justine Research Center in Montreal, Canada. He joined New York University, Abu Dhabi in 2014. Dr. Youssef's primary research interests are in understanding the role of genetics and environmental factors in shaping health-related phenotypes and how interactions between these factors can trigger disease and modulate outcomes. Today, Dr. Youssef will talk about genomic interrogation of the host immune response to infectious diseases in the context of malaria. Our second speaker is Dr. Ahmad Abu Tayoun. Dr. Ahmad is the director of Genomics Genetics Laboratory at Jalila Children's Hospital in Dubai, and also the associate professor of genetics at Mohammed bin Rashid University of Medicine and Health Sciences. Dr. Ahmad completed his PhD at Dartmouth College in the US. He then joined Harvard Medical School, where he completed his clinical genetics, molecular genetics fellowship, and in 2015 became board certified by the American Board of Medical Genetics and Genomics. Prior to joining Al Jalila Children's Hospital, Dr. Ahmad was the director of the Division of Genomic Diagnostics at the Children's Hospital in Philadelphia. Dr. Ahmad's primary research interests are in molecular diagnostics and genomics. Today, Dr. Ahmad will talk about using genomic approaches to understand clinical, molecular, and epidemiological characteristics of COVID-19, a very, very topical subject. Before we invite the speakers, or before we ask Dr. Youssef to start his talk, a couple of housekeeping issues I would like to bring up. All questions from attendees must be written in the Q&A box, and questions will only be answered during the Q&A panel discussion at the end of the two sessions. CME certificates will be available for downloading by tomorrow, inshallah, and this is the link. Finally, we also urge you to please complete a short survey at the end of the webinar. Without further ado, I would like to invite Dr. Youssef to present his seminar. Dr. Youssef, you have 25 minutes for your presentation. I will stop sharing the screen so you can upload yours. Okay, Dr. Youssef.
Can you hear me?
Yes, we certainly can. Thank you. Great. Go ahead.
Thank you very much. Thank you, Dr. Khan, for the introduction, and thanking also the Center for Genomic Studies for organizing this timely seminar on a very timely topic and very hot topic, of course, given the situation with COVID-19. So, can you see my full screen or not?
If you put it, it's not on full screen, put it on, that's it, yes.
Okay, perfect.
Excellent.
Okay, so what I would like really to start is really this, just have a reflection about how infectious diseases in general shaped human biology and human evolution through history. Here I'm showing just the life cycle of two pathogens. One is the Plasmodium falciparum, which is the causal agent of malaria, and the second is SARS-CoV-2. And the purpose of this slide is really to show the complexity of the life cycle of those pathogens. And as I mentioned, pathogens are a small fraction of all the microbes that exist on Earth, but they still cause a huge damage to life in general, plants and animals and humans included. And the level of interactions that we see between host and pathogens, it is outstanding in many ways. Malaria itself, it's a complex infectious disease. It involves two hosts. It involves various stages for the parasite during development. And it also involves all sorts of different forms that also can or has to adapt to the environment as well as to the host. SARS-CoV-2 as a virus, I would argue it's less complex than the malaria parasite, but still it involves a lot of interactions with the host. So infectious diseases have been the focus of studies for many decades, but genomics really revolutionized the way we think about infectious diseases, because now with the power of genomics and genetics, we can study these interactions between host and pathogens at different levels. I'm not going to focus in my talk on the pathogens, on the parasites or viruses or bacteria, but my main focus today is going to be on the host and how the host responds to infection. So genomics can enable a lot of applications. So if we're interested in studying the pathogens themselves, we can use genomics to understand the origin of the pathogen itself. We can try to figure out taxonomy. Is it a new pathogen or is it existing? Is it something that evolved? We can also develop methods of detection using genomics. We can do tracing, we can study the evolution, we can do prediction, how those pathogens will evolve over time. We can also use genomics to try help us with drug design. And that's for the pathogen. For the host itself, we can study other aspects. The main focus usually of research using genetics is to understand immune response specifically. And this is something very important, which relates also to vaccines, to development of vaccines, and also assessing the efficacy of vaccines. There are also a lot of polymorphisms in the human genome that really define how hosts respond to infection. And those polymorphisms can be different between individuals. They can also be different between different ethnic groups. And traditionally, for example, to be able to study the human host, in general, we use approaches like genome-wide association studies. This approach has been mainly applied to non-infectious diseases, but since 2007, the first genome-wide study on an infectious disease was published on HIV, and it was very interesting because it really revealed how variation in the human genome can affect how individuals respond to infection. And since then, we've seen many other genome-wide association studies like in TB, in leprosy. There are 2 genome-wide association studies on malaria. And more recently, we've seen genome-wide association studies on COVID-19. What you see here in this slide is just one of them. There is another major study that was published in December. And there are 2 or 3 other minor GWAS studies. And these studies have been instrumental to really pinpoint in the human genome where do we have genetic variation that can affect either disease severity or response to drug, or how we clear, for example, infection, and all sorts of phenotypes that relate to the clinic. Okay, so I'm gonna go back to malaria specifically, just really to give the rationale why we're interested in malaria. In my lab, we've been studying malaria for the last 6-7 years for a very specific reason. It's a global health problem. It's a complex disease. Malaria arguably caused a lot of deaths for humanity. There is a very interesting statistics that sort of estimated the number of humans who have been killed by malaria for 5 billion individuals. 5 billion of the 100 billion of humans who lived on Earth throughout the history of humanity. And malaria still kills a lot of people. And despite the drop in the rate of mortality in malaria, over 400,000 individuals still die every year from malaria. Just 15 years ago, it was 1 million individuals every year. And this is not a pandemic or an epidemic. This is something that is recurrent every year. And most, unfortunately, of those deaths are actually affect children. The majority are children under 10 years old. The majority are around, or even younger than 5 years old. Now, the major efforts have been put into the development of vaccines for malaria. Parasites are much more complex than viruses, so it's very, very hard to really develop vaccines for parasites. We only have one, there have been more than 30 projects to actually develop vaccines. All of them failed. The only malaria vaccine that we have is called RTSS, and it's the only licensed malaria vaccine with an efficacy that ranges between 26 and 50%. If you put this in perspective with what's happening with COVID-19, I think we should be very happy with the efficacy of the COVID-19 vaccines. Despite this relatively low efficacy, this vaccine have been approved, but we still see a lot of variation in the way children respond to those vaccination programs. So that was really the rationale for why we want to study malaria. Apart from the fact also that it's a very interesting disease for anyone who's interested in studying complexity and interactions between host and parasite and the environment. Okay, so now how do we use genomics in these studies? Genomics is an approach in itself. So it's the idea of really using as much information as possible to try to understand biological systems. And traditionally, in genetics, what we try to do is make links between genotype and phenotype. And we all know that it's a long way to get from a genotype to a phenotype. There is this black box in between the two, where a lot of things happen. Genes have to be transcribed. You also have the epigenome. RNA has to be translated. You have the metabolome. So there are a lot of things that can happen in this space between genes and disease phenotypes. And in the case of malaria, as I mentioned, it's a complex disease. So you have a lot of interactions between the parasite and the host. The parasite tries to hijack the host in terms of, for example, during the blood stage, the parasite tries to invade red blood cells to proliferate. And in parallel to what the parasite is trying to do, the host tries to fight it using the immune system. And really, to be able to understand infection, we should be able to understand both the parasite and the host. And this is the focus of my research program. So to be able to do this, we adopted a slightly different program than what is done in worldwide, which is to really focus on in vivo variation as opposed to doing experiments in the lab. And what we did, we set up a research program in Africa, in West Africa, in Burkina Faso. The reason for doing it in Burkina Faso, it's because we have a wonderful collaboration there, and also because malaria is endemic. So, and it's really a major health problem in Burkina Faso. Our design is a prospective design. So what we do, we go there during the dry season. We sample children before infection, before they actually get the infection. And then we follow them over time. And we sample children during the course of infection and after treatment. What I'm going to be talking about today is these 4 time points during the infection. And this is a design which is really important to be able to understand how the host responds to the infection. And we collect blood samples to do genetics. So we've done this for 2 years so that we are able to generate a discovery data for a discovery phase and then replication. So as I mentioned, we have a wonderful collaboration in Burkina Faso, and it's a lot of work to be able to do this. We work in rural areas of Burkina Faso. We work with a team of researchers and clinicians that are based there. We involve community leaders and nurses, and we have also a wonderful team that goes there from Abu Dhabi to be able to actually set up the protocols and do the sample collections. So I would like to thank them all for their contribution to this project. So now for genomics to be able to really take advantage of all the methods or applications that we can do, we collect blood samples from those children, and we try to really maximize how much information we get from those samples. So we've developed protocols in place to be able to really do DNA work, RNA work. We extract serum and also generate or isolate PBMCs to do cell-based lab work. And we've also been developing approaches and methods, both experimental and analytical, to be able to study a host and parasite at the same time. Now, just to give you an idea about the type of data that we are generating as part of this program, as I said, we're interested in using genomics in this systems genomic framework. So the idea is to really focus on mechanisms of response. And for this, we generate data on the genomes, the epigenome, the transcriptome, the metabolome, the lipidome, and also cytokine information on cytokines. The outcomes that we're interested in are severity, progression of the disease, clearance of the parasite, also looking at some of the symptoms. And our main phenotype of interest is parasitemia, which is how much parasite are present in the blood of those children, which is one of the proxies of severity of the disease. This is just a picture to show you how it looks under the microscope. You have one blood sample with high parasite load, so you can see that there are a lot of parasites invading red blood cells. And on the top, you see a blood sample with low parasite load, where we have less parasites invading the red blood cells. Okay, now going back to the main question that really intrigued scientists for decades, it's the fact that we don't have a vaccine, a functional vaccine for malaria. And the reason for that, it's the idea that the children, they are not able to develop specific immune responses against the pathogens. For an immune response to be specific and efficient, we need to have T and B cells, or what we call memory T cells and B cells, that recognize specifically the parasite to be able to mount an efficient immune response. Now, this is not something easy to do, to actually figure out what are those clones, those T and B cells clones, but genomics was really, really, really instrumental in helping us understand exactly the genomic sequence of those T and B cells to be able to identify the clones. So just to go a little bit technical here, this is again something that where genetics or genomics was very helpful. What we can think about, and here I'm using the example of T cells, you have a population of T cells, and each T cell is a different clone. It means because we generate this variation through recombination, and in the case of B cells through mutations, each T cell is different in a way. So by actually extracting DNA from this population of T cells, we can sequence each cell and then figure out exactly the sequence of the clone. And that gives us information about specificity of the clone. Why is this important? If you imagine the scenario here of a population of T cells, and imagine, for example, this specific clone is the antigen-specific clone, what you are hoping, if you are mounting an efficient immune response, would be that the antigen would interact with the T cell, and we would generate something that we call clonal expansion. You would generate, or you would expand the clone that is specific to the antigen. When this happens, usually you have an efficient immune response, and you will be able to clear the parasite. The same idea applies to vaccines. This is exactly what we train the immune system to do when we get vaccinated. Okay, so for us to be able to do this in malaria, what we did, we focused on a group of children, 10 children, and we followed them over the course of 2 years. And we sampled them before infection and during infection, again, in 2 consecutive years. And when we did this experiment, we generate a metric which is called clonality. So clonality gives us an idea about the expansion of clones within the individual. So, and this measure, as you can see here on the y-axis, the lower the value, the less clonal expansion that we see. And the higher clonality, the more likely they are clones that are expanding after infection. So when we compare the clonality of children before and after infection, you can clearly see that it goes down in both years. And even in the second year, more significantly than the first year. When we combine the 2 years, we see this trend of clonality, again, in general dropping. There are always exceptions. There are some children, for example, where we can see this increased clonality. But the trend overall is this reduced clonality after infection, which is exactly what we expected, given that, as I mentioned, for malaria, it's a complex disease, and children, specifically children, they don't respond efficiently to the infection. What's also interesting is this positive correlation between clonality and age. This is also expected because individuals build immunity as they age. When children survive beyond 10 years of age, they usually start building this specific immunity to malaria, which correlates with clonality as we see here. We also see a negative correlation with parasitemia, which makes perfect sense, because the higher parasitemia, the less likely you will have clones expanding that would fight the parasite. Okay, now I just want to show this to really show you that it's really complex. So I have to do things beyond group comparisons. Here I'm showing you two examples of two children. One children here on the left, where we're looking at the top 20 clones that are most abundant after infection. And we ask, are those clones expanding or not? It turns out that for this child, all of the clones are not actually expanding. And in the other child, it's the opposite trend. Most of the clones that we see after infection are expanded clones. Those give us hints about really how children respond to infection, probably driving this variation that we see between individuals. And the big question then is why? Why children are not able to develop this immunity? And the same question applies to vaccines. Why malaria vaccines are not functional and are not working? So we are currently doing a lot of experiments to really understand the mechanisms of why this is happening. Today, I'm going to focus on specifically one project that we recently published where we discovered one new mechanism of how parasites are using the host in a way to evade immune response. So in this project, and again, it's really using the power of genomics, we combine microRNA sequencing, total RNA-seq, and whole genome sequencing. And the design, again, is very simple. We look at groups of individuals over time before infection and then during what we call asymptomatic parasitemia. Those are children having the parasite in their blood, but they don't have symptoms. Then after a certain time, sometimes 1 week, 2 weeks, they might still have the parasites in their blood, and then they develop symptoms. And then we give them a treatment, and then we look at the parasitemia. And as you can see here, the majority of children respond very well to treatment. They clear the parasite, but there are still some individuals who don't clear the parasite. This is for our discovery phase. And then what we do, we look at the replication study the following year, where we focus only on the symptomatic individuals. Okay, so microRNA are tricky to work with. They're not like messenger RNA or total RNA. There are some extra steps that we need to do to generate RNA-seq data for them. Library preparation is similar to other methods, but there are some modifications where we need to actually run a gel. And here I'm showing you exactly the microRNA population that we isolate. So we actually cut the gel and then purify the microRNA, and then we construct a library. This is a bioanalyzer trace to show you that you have a very nice expected library with the expected size. And once we do this, we do sequencing. And through this, we generated data for about 320 microRNA. And here I'm showing you the dynamics of how the host responds to infection at the microRNA level. These are volcano plots. So we compare groups before infection, then right after infection during the asymptomatic phase, then transition to symptomatic, then after treatment. What you see here is that we have some microRNA that are upregulated and some microRNA are downregulated in blue. And what's really interesting is that this trend gets amplified during symptomatic parasitemia. You see that all the red that are here, they keep going up and all the blue keep going down. And what's also really interesting is that after treatment, we have the opposite scenario. So this really shows you that treatment works and that the microRNA profile in those children reverts back to the before infection state. What we do then, we look at the data from the second year, where we can, because we have parasitemia as a key phenotype, we can then ask a question, which ones of those microRNA correlate with parasitemia? And when we did this, to our surprise, the vast majority, except one microRNA, that are upregulated are also positively correlated with parasitemia. And then there are other ones that are correlated or that are downregulated after infection that are negatively correlated with parasitemia. So we use this combined approach to be able to really identify about 133 microRNA of interest. Those are microRNA that are responsive to infection or are associated with parasitemia. Now, well, once we found this, we decided to really look at the targets of microRNA. And that's why we generated total RNA sequencing data. And the hypothesis is very simple. MicroRNA, they target messenger RNA. And if we do a correlation between microRNA and messenger RNA, we should be able to identify microRNA and their targets that are negatively correlated. And the hypothesis is that if microRNA goes up, their targets should go down. And when we do this, we identify the number of genes that are negatively correlated, number of microRNA whose expression is negatively correlated with their targets. And to our surprise, most of those genes are actually involved in T cell function and specifically in apoptosis and cell death, as you can see in this enrichment analysis figure. So that really intrigued us because here what we discovered are microRNA that are targeting genes that are important, that are involved in apoptosis, and many of them drive those lymphocytes to actually die, as you can see here in this figure. Because we have lymphocyte data and cell counts, we see a very nice correlation also between this microRNA going up and the lymphocyte count going down. So it's like really we're here trying to close the loop between microRNA, their targets, and then the lymphocytes, and subsequently parasitemia. And we functionally validated one of those microRNA using cell lines. When we use a mimic microRNA, we see cell death in those cell lines, which is exactly what we see in vivo. So just in the last minute that I have, I want to also talk about another aspect that we can add to this analysis, which is a simple question. How does the genotype or the genome of the child affect their ability to respond to infection, specifically for microRNA? And for this, we do genome-wide analysis or EQTL analysis of microRNA and using whole genome sequencing data. And when we did this, we discovered 35 or 34 microRNA that are genetically controlled. It means that their expression is also controlled by the genotype of the child and not just by the infection. Here I'm showing you one example of a SNP that when you have the homozygote, you have higher expression of the microRNA. And what was really interesting is the association between genotype and parasitemia. So this SNP here that is associated with microRNA, it turns out that it's also associated with parasitemia. So if we simply ignore microRNA and do an association between SNPs and parasitemia, you can see this significant correlation between the two. And this microRNA, as I mentioned, is responsive to infection, and it's associated positively with parasitemia. So again, this is just showing you this proof of principle idea of how we can integrate different types of genomic data where we have a SNP, a microRNA, and a clinical outcome. And using what we call mediation analysis, we showed that this relationship between the genotype and parasitemia is modulated by this microRNA. And this opens a lot of venues for the idea of developing new ways of therapies. The details of these studies are published, and they are you can find them in this reference. So just as a summary, I hope really I convinced you that the genomics is a powerful approach to be able to understand host response to infection. We showed microRNA are responsive to infection. They are correlated with their targets and with lymphocyte counts and subsequently with parasitemia. And this really gives an opportunity for trying to come up with new ways to really think about infectious diseases. Here specifically, it has an implication on vaccine development because regardless of the vaccine and the efficacy, we have a mechanism that is driven by host factors that can affect the ability of those cells to respond to infection. So finally, I really would like just to thank the study participants and our collaborators in Burkina Faso and everyone here at NYU Abu Dhabi. And all the work that I showed today was done thanks to Isatu, Masar, Alpha, and Kiki in my lab. And thanks to also all the other members of my lab who contributed to this project.
Dr. Youssef, thank you very much. Excellent talk, and thank you very much for sticking to the time as well. Much appreciated. There's actually quite a lot of questions, but what we will do is, as agreed upon initially, we will try to address some of these questions at the end of both sessions. Now, I'll call Dr. Ahmad to present his talk, and he's going to talk about using genomics approaches to understanding clinical, molecular, and epidemiological characteristics of COVID-19. Dr. Ahmad, floor is yours. Dr. Ahmad, you're muted.
Thank you, Dr. Khan.
Yeah.
I hear you now. Thanks for the introduction, and I would like to thank the CAGS organizers for the invitation, and His Excellency also for organizing such a wonderful event, which is, I think, really timely, like Dr. Youssef had mentioned. So the title of my talk, as you can see, will be focusing on genomic approaches to understand COVID-19. And I think there hasn't been a pandemic or an endemic where the utility of genomics has been so powerful like we have seen in COVID-19. And to the extent that myself, who I'm not a virologist, I'm not an epidemiologist, but I work with a really great team of experts in those fields, where I try to work with them in using some of my genomic skill set to help kind of understand the epidemiology of the disease, and to see track mutations and look at host factors that might enhance our understanding of this pandemic. So I'll talk generally about the different aspects of genomic approaches that have been used, but I will also give examples using studies that we have conducted here in Dubai. Slide is not. Obviously, I have no conflicts of interest, so I might not have to do a lot of introductions about the virus here. It's obviously everyone knows this is an airborne respiratory pathogen, and because it requires the human cell to replicate, when we talk about COVID-19 genomics, we obviously talking about the virus itself and its genetic material, and we're also talking about the host genetics and any mutations in either the virus or the human cell that favors the propagation and the infectivity or the pathogenesis of the virus will help us understand how this virus induces its pathogenesis in order to be able to hopefully identify some ways of treatment or therapeutics. So I will start obviously by talking about the viral genomics itself. It's just hard not to talk about how genomics has been really powerful in terms of, from day one, actually, the discovery of this virus was done through a metagenomic approach. So if you remember, this has all started in December 2019, where there has been a number of unidentified pneumonia cases that had been linked to the seafood market in Wuhan, China. Those patients obviously had unexplained pneumonia, dry cough, fever, breathing difficulties, and some instances there were deaths. They were negative for non-respiratory pathogens. And so what happened back then is some researchers took nasal swabs or bronchoalveolar samples, and they extracted total RNAs from those samples and did shotgun metatranscriptomic sequencing. And those two papers that appeared in Nature in January, early January, basically looked at total RNA sequencing. And in this approach, they basically align all the RNA reads to the human genome, they subtract that, and then after this, they basically align the remaining sequences to a library of known pathogenic genomes. And in this case, the majority of the reads, after subtracting the human reference, aligned to this SARS-CoV-related coronavirus. And when they did phylogenetic multiple sequence alignment, you can see that this novel virus is aligning from those 4 patients to very closely to this bat coronavirus. And when you look at base by base alignment, which happens to have 30,000 letter long genome, you could see that it's very close to those bat coronaviruses, especially to this bat RATG13 virus with 97% homology, suggesting that its origin started from the bat. And so using this homology information and multiple sequence alignment and open reading frame predictions, we were able to basically predict the genome of this novel coronavirus We now know that it has 16 non-structural proteins in addition to 13 structural proteins, including the famous spike protein, envelope, membrane, nucleocapsid. I mean, the spike protein everyone knows about. It's this crown-shaped like receptor on the surface of the virus that's required for its entry into the human cell by interacting with the ACE2 receptors. And so when the virus infects, of course, the COVID-19 patients, it's going to replicate and generate millions of copies of its own self. And in the process of doing so, the virus will make mistakes or will have introduced mutations. Most of those mutations are generally benign and have no biological significance. However, sometimes they can be actually affecting the structure, function, or pathogenesis of the virus and might have implications for vaccine or any potential treatments. But in addition to this, those mutations actually serve as markers to be able to use in live in terms of tracking constructing the chain of transmission. And this is basically the crux of this genomic epidemiology approach that has been well used during this pandemic. And so the idea here is you would compare the genetic codes of different isolates at different time points, and then you do multiple sequence alignment to try to understand how those isolates relate to each other and what could be the source and the chain of transmission between those isolates. And this depends on two assumptions. One is the transmission chain. It's well known now that it takes around 7 days from the time a person is infected till they are able to transmit the virus. And it's also known that this coronavirus has a mutation rate of around 1 mutation every 2 weeks or 2 mutations per month. So if you put those pieces of information together, you can see that, for example, this blue isolate acquires a mutation every 2 weeks, becomes a new isolate, green, that differs from the first one by 1 mutation. 2 weeks later, it's now orange with acquiring a new mutation. So if you do random sampling and sequencing, and for simplicity here, I'm showing the genome as 4 letters, you could then look at sequence those viruses and then look at the mutations and try to infer that this isolate descended from this one and are two weeks apart. And then 2 weeks later, you have a new mutation. So in this sense, you are able to kind of construct how those viruses are related to each other and put a time frame to that. And so this is another hypothetical example. So assume we have 5 isolates. You could see that, for example, they all share this blue mutation. And if you were to construct a phylogenetic tree, this blue mutation should be at the bottom of the root of the phylogenetic tree. Then you have this orange mutation that will give up to all the 3 top isolates. And then you have this green one that will give the 2 A and B isolates. On the other side, you have this red mutation that will be the C isolate. And on the other side, you have this dark light blue mutation that is shared between D and E, and then this last red mutation that will be the E isolate. And so you won't do this manually. With some computational tools, you could do this and scale up to thousands, maybe even hundreds of thousands of sequences and align them and see how they are related to each other based on the sequence homology. And then based on this information and some metadata about where those viruses have been sequenced and probably some travel history information, you will be able to kind of construct a map of how the virus has been moving around and where has it been introduced and from which countries. And so let me give you an example of the work that we have been doing in Dubai, in collaboration, of course, with Dubai Health Authority and Mohammed bin Rashid University, Dr. Norbert Novotny, who's the professor of virology at MBRU, Dr. Sheikh Ali, who actually assembled the Dubai team, Dr. Hamdan Khan Sahib, who is the head of research at the DHA, Dr. Tom Looney, who's the epidemiologist at MBRU, and many other colleagues and the genomics team at Jalila Children's Hospital. So what we did basically, starting from January, as end of January till June 30, we tried to sample a representative cohort of COVID patients distributed by gender and age and having different presentations as defined by WHO, mild, moderate, or severe. And we have multiple objectives in doing so. So we ended up retrospectively recruiting 256 patients and mainly their nasal swab samples. We did RNA extraction for all 256 patients who we had clinical information about. Then we did two ways of sequencing. We did bulk metatranscriptomic sequencing, and we also did targeted sequencing for the COVID-19 virus, knowing the information and knowing the genome of the SARS-CoV-2 virus. And then we compared those sequences to each other and to also sequences that were published early on during the pandemic to understand where the virus came from in Dubai and the UAE and how things have been transmitting within the community. So our earliest study is we looked at the first 50 cases between the end of Jan and March, and using the phylogenetic analysis, we could basically see that 25% of the isolates had an Asian origin, and they specifically had a temporal entrance around the end of January. A third of the isolates had sequence alignment with isolates from the Iranian strains, and these appeared around mid-February. And then 50% of the isolates had sequence alignment to isolates that were sequenced in Europe, and these mainly started becoming more spread or prevalent towards the early March. And using this information, we're also able to kind of document the community-based transmission starting in early to mid-January. Obviously, when we extended sequencing beyond March, we could see that, as you could see, the three main routes, the Asian cluster, the Iranian cluster, the European cluster, but you could see that there are no additional routes or no main introduction, external introduction, but these preliminary or earlier introductions seeded additional clustering that basically each one of them just started expanding. And you can see that this expansion started towards the end of March, which overlaps with the closure of the Dubai International Airport to international flights back then. So you can see that the genomic data clearly overlaps with some of the public health measures, and that the introduction of the virus, at least into Dubai, has two phases. There's the initial external introduction with different time points, like I mentioned, from January, February, March, and there's strict community-based transmission starting the beginning of April. And then there's this, in that time frame, there's this D614G mutation that happened to appear in Europe towards the end of February and became the most prevalent mutation globally since then. So we were able to also track this mutation in our data set. It has been shown to be successful in terms of infectivity using viral load experiments and viral entries and cell line assays in cell lines. And so our data is also consistent with this global transition. I think my computer is taking a minute to switch the slides. But we also see the similar trend where the percentage of this mutation increased over time and reached around 70% of all isolates that we sequenced by June. So my computer is you can see my screen?
Yes.
Okay. Sorry, my presentation just went down. So this is what I just mentioned. So we do see a time dependent increase of the prevalence of the D614G mutation, at least between January and June early on during the pandemic. Like I said, this is, you could use those changes, mutations as marker to track the transmission of the disease, but obviously you want to understand the impact of those mutations on the biology of the virus and how it might affect its pathogenesis and potential impact on vaccines or treatments. So because we are doing nasal swabs and total RNA extractions and sequencing, and actually before that, we could actually, and because we have the clinical information about each patient, we could start doing some enrichment analysis to see if there are certain mutations in the virus that happen to be enriched in the severe as compared to the mild cases, and try to understand if some of those mutations in the virus are actually leading to more severe outcomes. And so over the course of this initial phase of the study, we sequenced 115 genomes. We identified 272 mutations. 20 of them were novel that has not been submitted in global databases. And we did this case or severe versus mild moderate enrichment analysis. Nothing is significant, which is known now that there hasn't been a single mutation that actually is associated with disease severity in the virus. We did see a couple of mutations in the same isolate in 2 patients, one of which is this missense mutation, the spike protein, reaching the 0.05 level of significance. And I think they were interesting because both were found in 2 young females that were otherwise healthy, but ended up having severe outcomes. The same mutation was also submitted in GSED, and when we reached out to the submitter, they shared their clinical information, and it was a young male who was otherwise healthy, but also was hospitalized due to COVID-19. So it's super rare. It might be deleterious to the virus, and so it can be associated with the severe disease, but obviously functional data would be needed to kind of validate this. So this is quickly the part about the viral genomics, and in the second part of the talk, I'll discuss the human or host genomics. Now that the main risk factors for severe COVID-19 is age. Obviously, the elderly are more likely to have a severe outcome. Males have more other comorbidities. However, of course, you cannot rule out genetics from a biological standpoint. The virus relies on the host cell to replicate and propagate, but there are also several cases where you see those young, otherwise healthy individuals that end up having severe outcomes and requiring intensive care treatments. And so the role of genetics is obviously there, and there are multiple ways where we can probe this. And I think Dr. Youssef has alluded to some of this in his initial slides. So you could look for rare monogenic variants that usually have large effects on their own. And the approach that you use to identify those mutations usually relies on less number of samples, where you do some segregation analysis, familial approaches, or do candidate gene approach. But there are the more common variants that have small effect sizes, and that in combination with other risk factors end up increasing the chances that an individual will have a more severe outcome. Such common SNPs are usually better detecting using large cohorts and genome-wide association studies. And like I said, I don't think we had a pandemic where we've been able to so quickly do a lot of viral sequencing, like hundreds of thousands of them, and also pick up on the host genomic analysis. And over the past year, not only we were able to track, to identify more infectious mutations, but have also been able to now identify loci and genes that are already important for the severe outcomes of the disease. And the first paper that was published follows this GWAS approach. And just maybe to simplify this, and there's a whole consortium, the COVID-19 Host Genetics Initiative at the Broad and Finland. So what they basically do is GWAS analysis. So assuming you're looking at a SNP, C or T, you genotype that SNP in cases versus controls, and you see the distribution of this allele. And in this case, for example, C is more enriched in the cases, so it's likely associated with the disease. But you obviously do this at a large number of SNPs, maybe 1 million or more, and you have a larger number of samples, and then you do this Manhattan plot where you look at those associations across the genome. And this, like I said, has already been done early on by the severe COVID GWAS group, where they basically recruited patients that have either severe COVID-19 or were controls. And they used 2 panels from Italy and Spain. Total numbers, I think, were 900 and 2,205 controls. And then their microarray had 8.5 million SNPs. Using this analysis, they found 2 loci that are associated with the disease. And this is a very powerful technique. It's more cost effective because you can snip more individual compared to sequencing, but it's an association study. It does not prove causality. So functional analysis are always needed to kind of prove which genes at these loci are actually the ones that are functionally associated with severe outcomes. Both are interesting. The first one on chromosome 3 has this chemokine receptors, CCR9 and CXCR6. And then the second one on chromosome 9 is the blood group locus ABO. And it showed that people with the blood group A are at higher risk of having severe outcomes, while blood group O is protected. But obviously, this is, in this cohort, they used a mixed group of COVID-19 patients. They were relatively old, above 60. They had other comorbidities, and so there is a mix of risk factors, and this is no surprise that the contribution of the genetic loci to the disease is a bit subtle. I think I'm still not switching. You see my screen?
Yes.
Right, so I think I'm having a hard time switching the slides for whatever reason. Let me see.
Dr. Ahmad, has your screen frozen or is on your side?
Let me quickly try to. Yeah, I think it's frozen.
Do you want to exit and load it up again?
Yeah, that's what I'm doing. So I think it sounds like my slides are very heavy.
That's probably what it is, yeah. Or there's some mutations going on in your slides.
Yeah, probably, right. I think this is my slide. I apologize for this. You still see my screen, right?
Yes.
And so you can see that, at each locus, the first one has an odds ratios of 1.7. The second, the ABO locus, is around 1.32. The second approach is looking for those rare monogenic deleterious variants in the host that might be associated or causing more severe outcomes. And this is the other consortium led by Rockefeller investigator. And so in this case, the assumption is, that most of the severe symptomatic patients are elderly or have other comorbidities. But what if we focus on the younger patients without any comorbidities that end up requiring intensive care units? And they've actually been very successful. They've just published recently this paper in Science, where they basically did the whole exome or whole genome sequencing on 659 patients with life-threatening COVID-19 and another 500 patients or more asymptomatic or have PCR positive but had asymptomatic or benign COVID-19. And they did a candidate gene approach where they looked at 3 genes that have been known to be associated with the pathogenesis in influenza, the toll-like receptor 3, and interferon regulatory factor 7, and a few other genes, but they focused on the rare, which are less than 0.1% for predictive loss of function variants in those 13 genes. And they found an enrichment of those rare predicted loss of function variants in their cohort, and those accounted for around 35% of patients with life-threatening COVID-19. They did functional analysis and in human patients, cell lines and in vitro, and showed they actually, disruption of those genes affect the interferon type 1 pathway and is likely to be the cause of severe outcomes. It's a great study. It seems like most of the patients are towards the younger age groups, but there's still a mix of still older patients, and therefore they had to sequence around 1,000 individuals, which is something you can only do when you have a big consortium like this. And so a better approach would be if you have families, right? So in this case that appeared in JAMA, there were only 2 families, 2 brothers on each side, and all the brothers were very young. I'm not sure you see my screen, but the ages were 21, 23, 32 years of age. They were had very severe critical COVID-19. One of them unfortunately died, and so they did whole exome sequencing in the 4 brothers, and combined together they identified a homozygous on the X-linked chromosome, loss of function variant in this toll-like receptor 7 gene, and a missense variant in the other 2 brothers. Functional analysis also showed that this toll-like receptor 7 also sits in the interferon 1 type 1 and 2 signaling pathway, and which also overlaps with the findings with this other study where they've sequenced 1,000 individuals. So it shows you how the approach really matters, and ascertaining the families with a certain in this case, there's 2 males, for example, both have severe outcomes, and just sequencing 4 individuals in 2 families was powerful enough to identify, to come to the same conclusions that this other paper did by sequencing more patients. Another way in my understanding of some of the genetics is really to focus on populations where you could look for enrichment of some of the mutations that generally are not associated with disease, but with the presence of viruses like this might bring people to the threshold of having more severe outcomes. And this is mainly something we could study in the Middle East because of the consanguinity. And the simple example here is that we all carry deleterious mutations, but if we have unrelated marriages, it's hard to have those deleterious mutations in the same gene in our offspring. But if we're related, the same rare deleterious variant will have a higher chance to be found in our offsprings. And this, like I said, can be normally non-disease causing, but with the presence of other environmental factors, such as the introduction of this novel virus, could lead to severe outcomes. So we recently received funding from Jalila Foundation to look for host genetic factors, especially in the young, less than 39 years old patients who are otherwise healthy but end up having severe outcomes. And we're in the process of recruiting those patients. We're targeting around 200 to 300 individuals for whole exome sequencing and transcriptome analysis. And finally, the last slide is when we did this bulk sequencing, we obviously have RNA from the human, from the host. So we are able to basically do host transcriptomic analysis from this nasopharyngeal specimen. And so we had only 50 cases that are stratified between mild, moderate, and severe that have enough good quality data to do this transcriptomic analysis. And the idea is again, like Dr. Youssef mentioned, is to find expression patterns that could distinguish the severe outcomes in patients with severe COVID-19 compared to mild, moderate, or controls. And this might give us some hints about some mutations or other factors affecting those expression patterns. We published this work And we did identify the usual suspects, in COVID versus control. We see high misregulation of cytokine, JAK-STAT, complement coagulation system pathways. All of these pathways are upregulated or are misregulated in all disease categories, the mild, moderate, and severe, but to a larger extent in the severe cohort, including specific genes and the chemokine and interleukin pathways. However, in this case, we had 37 mild, 12 moderate, and only 3 severe patients that we had enough good quality RNA sequencing from the nasal specimens to do this analysis, and obviously additional larger number of patients are needed to kind of validate those findings. And so to sum this up, viral genomics is really powerful, and we have already seen from day one, it was the way we actually identified the genome and the cryptic SARS-CoV-2 virus using metatranscriptomic sequencing. And after this, so many and tens of thousands, hundreds of thousands of this virus isolates have been sequenced to track how it's spreading globally and how certain mutations are becoming more infectious, and looking for mutations that might affect some of the emerging vaccines. But up to this point, there hasn't been any strong convincing data to show that there are mutations in the virus that are associated with disease severity. And that makes sense because most pathogens are naturally selected to be more benign so they can be able to propagate, so they can be more infectious, but they don't want to kill their host to be able to propagate. And then on the host genomic side, it's really all these studies and genomic technologies have been really amazing in terms of pinpointing using different approaches, GWAS or case control or familiar, at a very fast pace to identify candidate targets and genes that will help us understand the pathogenesis and that will uncover targets for treatments and management of this disease. And finally, I'd like to thank the hospital management, and specifically I would like to thank the Jalila genomics team, especially Satish, Maha, Ruchi, Yasmin, Noor, and Alan, who have been really working hard, especially Dr. Satish, and on the bioinformatics side, Dr. Ruchi on the transcriptomic analysis, and Maha doing a lot of the sequencing work, and our collaborators, and of course, colleagues at MDRU and DHA. Thank you.
Dr. Ahmad, thank you very much. It's been a busy session in terms of there's lots and lots of questions, to be honest, and it's virtually impossible for us to be able to address all of these questions. We will do our best to address. I'll pick up a few of these questions and try to ask the speakers to address these, and if possible, we may be able to forward some of these questions, and then the speakers may be able to address these to individuals later on. Just let me pick out at random a few questions, starting from earlier on. One of the questions was, this is, I think, to Dr. Youssef. Do you think that trained or specific TCRs, T-cell receptor in children, will be able to represent a robust memory phenotype during adulthood till aging?
It's a good point. It's the hope from vaccines in a way. Specifically in malaria, what we know certainly is that this memory builds with age, for sure. And arguably this comes from reinfections. Like when you have reinfections every year, you start building immunity. The parasite is much more complex than a virus in a way. So you can actually produce so many different types of T cells against different antigens of the parasite, keeping in mind that you also have the liver stage, you have the blood stage, you have gametocytes. So you have a multitude of possibilities to actually produce immunity against. Like I said, what we know is that this takes place with age for sure.
Thank you, Dr. Youssef. I'll throw one question to Dr. Ahmad. How can new variant strains affect the accuracy of the COVID-19 PCR test? And what should be taken in consideration when choosing the PCR test?
Obviously, I mean, it's a very interesting question. We don't do the test ourselves here at Jalila Children's, and we work with our microbiology experts at the Health Authority, and it's obviously that yes, if mutations arise where the primers are designed, you're likely to see dropouts and obviously false negatives. So. And this was one of the ways where some of the positive patients turned out to have this S dropout for the UK variant because of this deletion. So yeah, definitely mutations are likely to affect primer binding and the accuracy of the PCR test. And therefore, I think it's really important that we have these genomic surveillance programs that are active, especially in the UK as a prime example, where they're sequencing more than 3 or 5% of all positive cases. And that's how we're identifying those cryptic mutations and strains that will further understanding and better choosing the proper sites for the PCR tests. So it definitely affects the accuracy and should keep an eye on that.
Dr. Ahmad, I mean, just a very interesting, very, very quickly, I'm kind of desperate to ask that question because it's following up from this, and I think another time is limited. The variant strains, specifically the UK strain, and now more recently the South African strain, which seems to at least there's indication that it may be resistance to the Oxford AstraZeneca vaccine.
Yes.
What's your take on that? I mean, is there a danger that some of these strains, we may get an emergence of a strain which is actually resistance or to the vaccine, and the vaccine no longer cover?
Yeah, and I think I can answer from so most of the Oxford, the Pfizer, the Moderna vaccines they don't use the traditional way of making this the inactive virus, like for example Sinopharm, where you have the whole virus injected into the vaccines. You're likely to make antibodies across the whole virus, including the spike protein. But for Pfizer, Moderna, and AstraZeneca or Oxford, you're basically engineering the segment that codes for the spike protein, and you're likely to make antibodies against this specific region. And so mutations on the spike protein that might affect the binding of the antibody or the neutralizing or antibodies against this are likely to, those mutations are likely to disrupt those interactions and therefore might affect the efficacy of some of those vaccines. I've seen some of those studies, but my understanding, for example, the Moderna and the Pfizer, that some of those strains reduce the titers, but still we have enough that will be safe. But in the case of AstraZeneca, and what I saw and I think is that it's actually might be close to affecting the efficacy of that virus, again, because we're relying on the spike protein, and changes on this will likely disrupt. And now we're putting pressure on the virus. We're adding all these vaccines, so we're naturally selecting for things that are likely to escape, right? So again, it's really important to keep an eye, especially on the individuals post-vaccination and at population level, see which strains or isolates are continuing to spread around after we vaccinate the majority of the population, so we better understand what's going on.
Thank you. Dr. Youssef, there's a question asking why how cytokines impact mRNA in microRNAs in parasitemia.
So the short answer, we don't know specifically. The link between cytokines and microRNA, it's not very clear. Cytokines can affect cells that produce RNA, but again, at least in the context of malaria, we really don't know.
There's a question. COVID obviously is a hot topic, so I think there's a lot of questions with regard to that as well. There's a question: what is the possibility of recombinant virus which may result from a person who's co-infected with MERS corona as well as COVID-2?
I might want to defer to a virologist. Maybe I don't know what the chances you'll get those two together, right? And then theoretically, I mean, this is now an emerging hypothesis that, for example, the UK variant has emerged through a recombination event that just gave up those 8 or more mutations in one go. Because it sounds like, when the virus is infecting a certain host, it's diverging. And so having those divergent isolates within the same host, and then each has its own mutations, when you recombine, you make up those a bunch of mutations that since this virus has a huge playground, it's a global one, it's not a fast mutating one compared to the influenza or HIV, but it has a huge landscape that the events could happen that even if it's rare, that you'll have this recombination event that will be really highly infective, like the theory for the UK. The question of MERS and SARS-CoV-2, I'm not so sure how often that will happen theoretically, and also you have to take into consideration how homologous the two genomes are to be able to come together and do the recombination. So,
well, we know certainly that this happens, of course, in influenza. Well-known example is influenza. We have recombination happening in influenza. This is how we get pandemics. I assume this is why this is on the basis on that the questioner may have asked that question. Question for you, Dr. Youssef. Thank you for a very interesting talk. How do you mechanistically think that the vaccines might mimic parasite response in upregulating specific microRNAs you found? And do you believe that they can be modified in a way to heighten the T-cell memory response?
That's a good question. Again, this is something that we're currently investigating because we actually don't know the mechanism of why right after infection, certain microRNAs go up and some go down. That specific point, we don't know. We have to do a study that really looks at the vaccine response. And this also applies to COVID-19. You can really look at natural infection, see exactly what's happening, what's taking place. You can do exactly the same thing with vaccines. You would just need like some sort of randomized trial where you would vaccinate some people and others don't, and then look specifically at, do we see the same types of microRNA going up or down? If that happens, that would be very interesting. It would really show that there is something mechanistically that really is present in the vaccine that is also present in natural infection that triggers those microRNA. If we understand that, then we really can take it to the next stage, which is therapeutics. And the microRNA have been very promising as new ways of therapies, because if I can block or use microRNA itself to trigger something that I don't want, that can actually give hope to limit the undesirable outcome.
One more question. I think we're running very, very short on time. What I will do is ask just one more question each. There's a question here about blood groups, whether blood group O is protective, whereas blood group A is affected by COVID-19. Why is blood group A protected? That's what the questioner, I think, is asking.
Right. I mean, that study is, like I said, it's an association. It does not show the functional, like the mechanism. But I think in that paper, I think what they speculate is, because the viruses have those glycans on their surface, that might be, similar to the glycans on blood group A. And if you are blood group A, you won't have antibodies against those glycans on the virus. While if you're on blood group O, you don't have the A antigens, so you're able to generate antibodies against those glycans. And therefore, that's why O might be protected versus A is not. I think that was one speculation, but I don't think I have seen a functional study that kind of, at least to my knowledge, that has shown how mechanistically that A is more susceptible to severe disease. By the way, not COVID. They still can get COVID-19, but the blood group A are more likely to get severe outcomes, especially respiratory failure, compared to blood group O, who if you get COVID-19, you're more likely to be protected against respiratory failure.
Dr. Ahmad, there's a very related, I did say it's the last question, but there's a related question here that was saying that how is gender affect the infection and with corona? So I assume that they're talking, we already know that, of course, males are much more likely to have severe disease compared with females. And I assume what the question is asking is do we know why males are predisposed to more severe disease than females?
Yeah, and I think, again, to my knowledge, is the hypothesis is that males have the higher expression of the ACE2 receptor and are more likely to be taking uptaking at least the viral loads at higher level. But that's my my only thought, if I could let that be.
Dr. Youssef, same question. Is that, I think in terms of immune response, I think I came across some literature recently with regards to cytokine expression with regards to the difference between males and females. Are you aware of any differences in terms of the immune response to COVID-19 between males and females?
Now, I've looked at the literature, and there is also a lot of because there is also behavior which is associated with gender. There is confounding in terms of exposure as well. So I think there is no really strong evidence apart from what Dr. Ahmad mentioned, which is the expression of the ACE2 receptor. There is a lot of confounding factors with age, with gender, that should be considered in this. There is also the immune response, as you mentioned, cytokines specifically. We know those differences exist between males and females, for sure. Some of them are also linked to age. The most controversial that I've seen was this association even between different ethnic groups. Like you have some variants that are more prevalently, say, in Italy or in China, that are also can drive some of these differences that we see.
One last question, and that's actually a question from me, something which, again, is something which sort of occurred to me in terms of one of the attendees asked a question with regards to vaccine for malaria. Some years back, if I remember right, some Australian groups and a number of other people, I think they've tried using, if I remember right, Wolbachia, one of the bacteria, and infecting mosquitoes with this bacterium. And what the studies show, I think these were in Nature or Science, and what they showed was that parasite infected with, or the mosquitoes infected with the bacterium prevent the replication and hence the transmission of various viruses, including Zika, if I remember right, and dengue. Could that be and they actually released these mosquitoes into the population to become the dominant type. My question is, could that really work in terms of preventing malaria as well? Could we do that?
It's a very good question. There is so much that we tried with mosquitoes, and they're really, really resilient. I mean, there is even CRISPR trials in Africa to actually CRISPR the mosquitoes or even attempts to CRISPR the parasite itself. But again, if we learn something, is that those mosquitoes are very, very resilient. They develop resistance to drugs. There is also, I should mention, the issue of climate change. And this is really a threat in the next decades, because the more climate change with global warming, the more expansion we're going to see for those mosquitoes. So many countries that have not been exposed to the mosquitoes that have been declared malaria-free will be at risk because of climate change. So again, there are so many avenues that scientists and clinicians are working on. Some target the mosquitoes, some focus on the host, some focus on the parasite. And we really need a global approach to be able to solve this problem, at least in malaria. The last thing I would like to mention is we have so much to learn from malaria and other infectious diseases in this fight against COVID-19. So many methods that have been applied to infectious diseases in general can actually be very useful in fighting COVID-19.
Dr. Youssef, Dr. Ahmad, thank you very much for an excellent talk. And again, apologies to some of the attendees who asked there were so many questions, but we could only address limited number of these questions. Finally, what I would like to say is, please do fill in this short survey at the end of this seminar. The CME certificates are available for you to download. Hopefully these will be available by tomorrow from this website. And that's also been communicated to the attendees through the chat box. And finally, our next webinar is on population genetics. And this will be provisionally scheduled for the 24th of March. But there will be a series of these seminars going on throughout the year, organized by the Center for Arab Genomics. Finally, I would just like to thank the hosts, the speakers, and all the attendees. Stay safe and see you next time, inshallah. Take care. Assalamu alaikum.
Thank you.
Thank you.
Thanks.